screen. As our colleagues join, maybe we can give them a minute or so. Any questions from last time? I think we spoke about influenza, human influenza viruses. We talked about the family being Orthomyxoviridae. Interestingly, the first two viruses in today's session are in a similarly named family called Paramyxoviridae, and we'll be looking at the differences. We also looked at antigenic variation. I hope you still remember what that was. And we looked at the options, treatment options. Influenza is one of the few viruses against which we have specific therapies or therapeutic agents. And we also looked at the vaccines and other forms of deterrence and prevention. Somebody has asked why acyclovir cannot work on influenza viruses. Anybody would like to answer that? I think it was a direct message, so I'll just copy it and paste it in the chat. Why can't acyclovir work on influenza viruses? Anybody? Songok says acyclovir is for DNA viruses such as herpes. And yes, acyclovir targets the DNA polymerase specifically for herpes viruses. There are other DNA viruses such as hepatitis B, human papilloma viruses, adenoviruses. These are not amenable to, or they are not sensitive to acyclovir, even though they are DNA viruses. So acyclovir is specific to herpes viruses, and even for herpes viruses, it's not all of them. Which herpes viruses are sensitive to acyclovir? Anybody? Somebody is talking about HSV2 and HSV1, and yes, any other? HSV1 and 2. To some extent, varicella zoster virus as well, which is human herpes virus 3, but not CMV. CMV is not sensitive to acyclovir. We would have to use the acyclovir or other agents. So it's good to know specifically where these agents work, unfortunately, because there's no blanket. We would like to have that one magic bullet which can treat all viruses, but I don't think we have one yet. Maybe one of you is going to develop that. So I'm going to start now. As I said, our topic is measles, mumps, and rubella. They cause similar diseases, and that's why they're categorized together. They're not the same viruses, but because they cause, it's like hepatitis viruses. They come from very different families, but they cause a similar disease. So
class. 1 second.
What do I have? Thanks for muting everybody. There are some guys who are in a dance floor somewhere. The paramyxoviruses, I would like to introduce this family because we are learning about 2 of its members today, and you're also likely to cover others if you haven't covered already with Prof. I think Oyugi. And this is a family in the order known as Mononega virales, which we talked about when we talked about rabies. And these are single stranded monopartite negative sense genomes. Monopartite as compared to segmented. And you can see the viruses are also enveloped And interestingly, these ones have two surface proteins, just like orthomyxo, but the surface proteins are different. In this case, you have a fusion protein. Remember, influenza had neuraminidase, neuraminidase and hemagglutinin separate. But in this case, you have an attachment protein, which is either hemagglutinin or has hemagglutinin function. It has hemagglutinin and neuraminidase function together, or has none of the hemagglutinin or neuraminidase function. And in that instance, it's called a G protein. After some classification in the last few years, most of the G have actually been put in a different category. But the important viruses that have no hemagglutinin or neuraminidase function were mainly RSV and metapneumovirus. RSV is respiratory syncytial virus, and it is very important for you to learn about it because it can cause death in children who have viral pneumonia. And it's no longer part of this family, but it's the one that one of the viruses that has this G, which means it has no hemagglutinin or neuraminidase, but all of them still keep the fusion. And that's why you hear a name like RSV involves syncytial formation because of the fusion of the host cell membranes when the virus attacks adjacent cells. So in this table, I've just summarized a few genera which are relevant. There are many others. I think this family has more than 15 genera or thereabouts. And the first one is human respirovirus, respirovirus genera, which contains human respirovirus 1 and 3. We know them better as parainfluenza 1 and 3. And you can see these have both hemagglutinin and neuraminidase function in the attachment glycoprotein. And as you see, all of them, as I said, have a fusion protein. The second family, we are not going to discuss genera 1 today. The second genera or genus, sorry, is Rubella virus, and this is where Mumps virus belongs to. The full name for Mumps is actually Mumps Rubella virus. But you also have other parainfluenza viruses 2 and 4 in this genus, although these have been renamed from parainfluenza virus to Rubella virus 2 and 4. So as you read text and as you read even some of the older presentations we shared, you will see these names, the older names, human parainfluenza viruses. But moving forward, ICTV, which is the International Committee on Taxonomy of Viruses, has reclassified these viruses, and they keep doing this as you get more information on the viruses. And so this is sometimes can be frustrating to a student because when you're reading today and maybe next week there's a categorization, something changes, sometimes can be a bit confusing. So we are not too hard on the new changes, but it's good to know what is happening in the field. If you confuse them a bit, it won't kill you, but you need to remember at least the virus names. Morbillivirus is an important genera or genus because we have measles in this group, and we also have a similar virus called rinderpest. Now, rinderpest is not a human virus. It used to cause what was called as cattle plague or plague. It's actually one of the suspected to be one of the plagues in Egypt. The rinderpest was eradicated, and it's the only second disease to be eradicated in 2011. And this was declared so by the equivalent of WHO, which is Food and Agriculture Organization, and it is a disease that was causing a lot of death. Fatality rates were about 90%, so it had a lot of economic and food security issues. And it's very similar to measles, and because it was eradicated, that should encourage us that we can also eradicate measles. And in fact, rinderpest had more hosts than measles because it could affect what we call even-toed ungulates like cattle, buffalo, antelope, and what have you. Henipavirus is a zoonotic virus, and usually in bats, and you can see, you may have seen reports in India and a few other places, and you can see it's one of the viruses without hemagglutination or neuraminidase function on its attachment glycoprotein. And it's also one of the viruses you are likely to cover with Profoyugi when you're talking about rodent-borne viruses. So in short, Paramyxoviridae is a family with very many diverse members. In fact, the entire family includes viruses that infect reptiles, birds, and what have you. these are the ones, the general I've listed here, the ones that infect humans. And we are going to focus on mumps and measles. And just from this table, you can see that mumps has HN, or both hemagglutinin and neuraminidase, while measles only has hemagglutinin. If you see clearly on this table, we do not have rubella virus, because rubella is not a paramyxovirus. It belongs to another family, and we'll discuss that family when we get to it. So we can get into measles now, unless there's a question. And measles is, oh, I see somebody asking about why rhinovirus is naked and yet acid liable. Enteroviruses are not generally acid liable, but rhinovirus doesn't seem to be causing a lot of gastro diseases. So it's an interesting question. Maybe you can tell us why you think it is not acid liable. But most enteroviruses will be gastric acid resistant. Does rabies virus also have non-segmented genome? Yes. The mononega virales have a single partite genome. This includes Rhabdoviridae, include things like Ebola, which are in Filoviridae. So the viruses that are segmented will actually be mentioned specifically because they are few. And this, so far, we've talked about influenza viruses. I don't know if you have talked about Rift Valley viruses, which is in a larger family, actually not a family now, but a class called Bunya viruses. So that is the second group. And the third group that you may come across is rotaviruses, and this cause diarrhea in a family called Reoviridae. So there are not many segmented viruses. Most viruses will be non-segmented. Now for measles, and just for emphasis sake, we've indicated before that it is in the genus Morbillivirus, which has both fusion, has two surface glycoproteins. One is F for fusion, and the other is H for attachment. It is a single-stranded negative sense RNA linear genome, which is enveloped. And remember, enveloped viruses are easier to disrupt because detergents and other elements can easily destroy the envelope, making the virus unable to infect. And the virus used to infect nearly everybody before the vaccine was introduced in 1963. So it was very common, and the vaccine changed that. Transmission is usually through aerosol or direct contact. It's highly contagious, with more than 90% of susceptible contacts being infected. What was the rate for HIV? For HIV, you have about 8,000 infections for every 1,000 contacts. 8 out of 1,000. That's quite low. This is 9 out of 10, or 900 out of 1,000. So you can see the difference. Man is the only host, and there's no animal reservoir. And this should help us in eradication of the disease. So when the man is the only host, it means if we give all human beings and their babies the vaccine, then nobody will be having measles. So then measles can disappear. But we haven't achieved it yet. Maybe you guys will tell me why. Infection leads to lifelong immunity, or even successful vaccination. I say successful, we will discuss that later in the vaccines, what that means. It leads to lifelong immunity. Although, of course, we know there are people with immunosuppression, like HIV and other issues that may may interfere with the immune response and production of antibodies, and even cellular immunity sometimes, because remember, antibody response also relies on T helper cells. So that may also take away the immunity. In terms of virus structure, it's a very simple virus, as you can see here. As we described the virus before, it's basically a genome packaged and protected by protein envelope. And this linear genome here, you can see, is surrounded by a capsid protein, and you can see it's associated with the L and P protein. And just to jog your memory, with rabies, which is another mononega viral, we had L being the polymerase, and then the P is a phosphoprotein, which work together during replication. But also the phosphoprotein has an impact on the immune system. It suppresses the immune system. So this is one of the similarities between the mononega viral measles and the mononega viral rabies, which are in different families, but in the same order. The other proteins here are the matrix, but the other important one is the H protein and F protein. Remember, when you see H, that means it can cause hemagglutination on red blood cells outside the body in a petri dish, not in the body. F protein is basically for fusion, and what that means is that if the virus has infected one cell, it can recruit nearby cells by just fusing the membranes, and that avails more material for the virus, more DNA, RNA bases, more enzymes, more protein synthesis mechanisms or structures without having to leave that cell. So it basically just emerges nearby cells, and it helps for the virus to spread without having to leave a cell as such. Doesn't mean doesn't leave, it can, but fusion helps it spread. Measles epidemiology is, as I said before, we've seen worse days, and because of the transmission, which is very easy and also highly infectious, a lot of people used to be infected, nearly everybody, and used to have hundreds of thousands of deaths, but in 2018, 140,000 deaths were recorded, which was equivalent to a three-quarter reduction since 2000. So the measles vaccine has been touted as one of the most effective public health interventions because it has reduced or done away with millions of deaths in the last 2 or 3 decades. Unfortunately, most deaths occur in children under 5, and that's why we try to emphasize a lot to give these children and protect them, vaccines, and protect them when they're still young. Pathogenesis. Infection will begin in the upper respiratory tract. So this means the nasopharynx, the oropharynx, and then it will spread to the local lymphatic tissues or lymph nodes in about 2 to 3 days to 4 days. From there, of course, in the local lymph nodes, it will multiply. And then there is viremia, which disseminates the virus to other tissues, including the skin, where we see a rash, which is thought to be a type 4 hypersensitivity reaction. This is a delayed type hypersensitivity reaction, which is cell-mediated, meaning that infected cells will be attacked and destroyed by the human T cell, killer T cells. And as the virus spreads, it will seed in many other organs, not just the skin. So it may have different effects depending on the organ that is most affected. Remember the protein that we mentioned before, the phosphoprotein that suppresses the immune system. That immunosuppression can lead to vulnerability and even infection by both bacterial and other viral infections, things like streptococcus pneumonia or staph aureus, or even adenovirus pneumonia, because the body's immune system has been suppressed and the epithelium has been affected, is not clearing very well the pathogens. And this can cause bronchopneumonia, and is actually a common cause of death in children. Now, rarely, you may have the virus itself attacking and destroying the lung cells. And this could be the interstitium, could be the pneumocytes. And by that destruction, remember how the virus attacks cells and then fuses the membranes, forming giant cells, like some syncytia of some sorts. And this can cause pneumonia, of course, because every time you destroy cells, you cause inflammation, which causes leakage and recruitment of more cells. That causes debris to accumulate, more immune cells to try and fight the virus, and that causes accumulation of debris and fluid in the lungs, interfering with oxygenation. Fortunately, this is rare. And if somebody recovers from all this, they develop lifelong immunity, unless, of course, there's a reason for immunosuppression later. And immunosuppression can come from infections like HIV, can come from diseases like cancer, treatment for diseases like cancer and things like that, even steroids. The clinical presentation is usually very, I would say, it's distinct in a way, although it has differentials. Incubation period running from 10 to 12 days, when then the person develops a fever, which is called a high fever, basically usually more than 30 degrees Celsius. They may have this classic triad, although don't insist that they have it. If they don't have it, they could still have measles. But the classic triad of measles is cough, coryza, and conjunctivitis. basically a cough, a running nose, and red eyes, inflamed eyes. That is the conjunctiva. Koplik spots are pathognomonic for measles, and these are usually, I think I'll show an image on the inner side of the cheek, on the back of mucosa. You may have, next to the teeth, you may find some grayish spots there. And usually they may come just a few days, a day or two before the rash. And they are pathognomonic. So if you see that, you know that person has measles. Rash will occur about 10 days, 14 days after exposure, and it will start with the head and then spread to the trunk and then to the leg, to the limbs. And it will last about a week. During that time, the person will be infectious. You can see here some images of the Koplik spots on the buccal mucosa, and you can see the teeth are not too far. And this person has opened their mouth, of course. The rash usually appears after this, maybe about a day or two after the Koplik spots. So if the child is coming to you with a rash and the rash has lasted 4 or 5 days, you may not see these Koplik spots. But if the rash has lasted about 1 or 2 days, you may be able to see them. It's unlikely that a mother will bring their child to hospital because of these Koplik spots, unless, of course, the fever might bring the child to you. So if the child has had fever for 1, 2, 3 days, and you find these spots, then you may suspect measles, even if they don't have a rash. This is what we call a generalized maculopapular rash. You can see it in the entire body. This is on a darker skin, and you can see the macules and papules. Basically, some are raised, some are flat, some are wide, some are pinpoint. And more dark skin, and this is all maculopapular rash. Some people call it a mobiliform rash. Complications can occur as well. One of the common ones which causes issues is bronchopneumonia. Because of the immunosuppression, the destruction of the mucosa or the epithelium, children or people with measles, generally, most of them are kids anyway, tend to have bronchopneumonia. So you need to keep that in mind. And for that reason, look for symptoms of pneumonia and if necessary, even treat. Otitis media can also occur, which is usually, remember our inner ear, middle ear is linked to our respiratory tract. So these two are connected. You could also have an inflammation of the upper airways, and in children, this tends to cause croup because the children's airways are narrower than adults, and any inflammation can cause obstruction, and this can actually be life-threatening. Some kids may develop diarrhea, blindness, and even encephalitis. Encephalitis can be acute, subacute, or chronic. The chronic form is called sclerosing. This is a misspelled word, sclerosing panencephalitis. And sclerosing panencephalitis or subacute sclerosing panencephalitis is rare, usually just 7 in a million cases of measles. And it can develop years after the infection, even 10 years after. But once it develops, it's progressive and it's usually fatal. It cannot be really reversed. Unfortunately, there are not so many of these cases now that are being seen and studied, so we may not be able to do additional research that it can be meaningful. But we know if we give everybody vaccines, then we'll be able to stop it. And usually survivors will become severely disabled physically and mentally. Usually it will begin with behavioral changes leading to motor abnormalities, things like ataxia, speech, and then issues with swallowing, and even vision may be lost or impaired. And this can last for weeks to years. And then person eventually end up with death. How do we diagnose measles? Let me see if there's a question. What does epidemiology and pathogenesis mean? Oh, this is actually a very good question because it's common in exams. So if we ask you about pathogenesis, we're asking you to think through the process that the virus will cause disease. So for example, in a single line, measles can cause disease by destroying cells directly, that is direct viral effect, or by triggering the immune system to destroy infected cells, or by suppressing the immune system. That is in a nutshell. But you need to take us through that process from infection. Infection could be, how does it get into the body? After it gets into the body, where does it go? Like measles, we've seen it goes into the lungs, then there's some viremia, dissemination to the rest of the body. With influenza, we saw it only goes to the upper respiratory tract and sometimes to the lungs with no viremia. So pathogenesis, think about how does the disease cause symptoms. Don't just list the symptoms. In fact, if you just list symptoms, you won't get any marks. Think of how do those symptoms come about. So for example, I don't know if you've learned about polio. If somebody is polio presents with paralysis, if we ask you to describe pathogenesis, you should tell us how that polio got into the body and how it led to paralysis. That is pathogenesis. Epidemiology is a lot about transmission, distribution within a population, within the world, and also a lot about issues like prevention and all that. So it's difficult for us to just ask you to describe the epidemiology, but if we just ask you to describe the epidemiology, you can look at very broad aspects, from transmission, risk factors of infection, to prevention, to distribution of the disease, and how many people get sick and die and all that. But pathogenesis is about how the virus causes disease or how the pathogen causes disease. How, not which symptoms. And also some people get lost in replication cycle. It's not really the most important thing under pathogenesis, but we would like to know where the virus replicates. You don't have to describe the entire replication cycle. For example, if it is polio, if it initially multiplies in the enterocytes or the cells lining the small intestine, and then through circulation goes into the anterior horn cells where it replicates further. Those are details we'd like to know for pathogenesis. Because you see, you get hepatitis B through sexual contact, and yet nothing is wrong with your genitalia when you have hepatitis B. The problem is in the liver. So the question comes, how does this hepatitis B virus end up causing jaundice? And if you understand that pathway, then you've understood pathogenesis. Okay, so diagnosis, I hope that's clear. You can prevent SSPE by giving the vaccine, because you don't, if somebody does not get measles, then they will get SSPE. Once they get measles, you can't do anything. You just, it's a game of chance. Fortunately, it's rare. A prodrome phase, usually the fever and the non-specific symptoms are the prodrome. They're basically just non-specific symptoms that don't necessarily have a specific presentation. It's like what we call constitutional symptoms. I see another question. Oh, coryza. Coryza, simply put, is when you have a running nose, but it involves inflammation of the nasal mucosa and all that. So the best way to put it is a running nose. Are we required to know the mechanism of all drugs? Yes, 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 because there are not so many. And we have a session for it. But for each category, for each virus you study, look at the specific antiviral agents and the mechanism of action. It's actually the best way to remember antiviral agents. If you try to remember all of them, you may not, especially when you get to things like hepatitis C. So remember the mechanisms and maybe an example for each mechanism. So yes, that is expected. Cytopathic effects and examples, I'd like to read that, but generally is what the virus does to a cell. Some of them will lyse the cell, they'll destroy it. Others will, because of inclusion bodies, others may have, if you look at a microscope, different effects on these cells, but the main common one is lysis. Some may even transform the cells to malignant ones, but that is called transformation. Now, diagnosis of measles, one is clinical. As we said, you may see, especially in that age group of children, a rash or sometimes the complete spots with fever, and really that is it, rash and fever, not much. We said coryza, conjunctivitis can occur and cough, but they are not a must. They could be, especially when a child is partially immunized or has some antibodies from the mother, but they're not sufficient, they may present with a typical disease. Lab diagnosis will include serology, and for serology, we usually look at IgM antibodies. So if for you guys right now, if I take your blood and I find that you have IgM, it means you've been exposed to measles recently, and you even may be sick if you're not immune. But if you have IgG, and I've only done one test, I'm not surprised. Even me, I expect to have IgG, and I'm healthy. I would like to think so. So what does that mean? IgG is just a marker of exposure previously, and the exposure could be infection or vaccine. So for you to show infection, you need to have a paired IgG measurement. You take an IgG measurement today and maybe a week later, which shows a fourfold increase. But if there's no increase, that IgG does not mean infection. It just means actually protection. Because if you have IgG against measles, you are protected. PCR, usually from the throat or nasopharyngeal swabs, blood or urine, will give you a definitive answer to the virus, although many times we don't do that. And the culture can also be done on the same samples mostly, but not so much on blood. These usually are for research purposes, not for diagnosis. The treatment is supportive. There's no specific treatment for measles, and depending on what the child needs, you learn a lot about measles treatment in PIDS. It includes a lot of things from rehydration, antipyretics, even vitamin A supplementation and all that. And this is really supportive. Some people have tried to use ribavirin for SSPE, but there's not much success with that, and we don't have enough numbers anyway to show a good powered study to show that. Control is usually through vaccination, and sometimes you can give post-exposure prophylaxis with immunoglobulin and the vaccine. So what vaccines are available? In public health, these are called measles-containing vaccines. They are live attenuated virus vaccines, which basically means a virus that has been cultured, weakened, and then is prepared to be given. You can give a measles-only vaccine, which is what a lot of us got as kids. Now we are giving measles and rubella vaccine in Kenya, and this is done at 9 and at 18 months. I'd like to see in the chat why you think we need a repeat at 18 months. Before we didn't need it, or before we didn't give it, I think we still needed it. And in fact, in some babies, you can give it at 6 months. So why are these differences? Why do we give it at 6 months for some? But generally for others, why do we need to repeat at 18 months? You can also give measles, mumps, and rubella. This may be available in some private hospitals, but the government ones is just measles and rubella. You can also have measles, mumps, rubella, and varicella vaccine. And all these are live attenuated vaccines. So any idea why we are repeating at 18 months? I see a question here on is the serology similar to herpes? EBV, where presence of IgM shows current infection? Yes, IgM shows current infection. And a lot of us have been exposed to herpes viruses. So if I take your antibodies, I may find at least you have 1 or 2 or 3. Some of you even have these cold sores which you blame malaria for, but it's actually herpes. IgM receptors are found in basophils, or I don't want to get into that. This is not immunology. Maybe maternal antibodies have weaned off. I think this is a response to one of my answers. At 18 months, when the mother's antibodies are out. So yes, at 18 months, we want to make sure that the child no longer has maternal antibodies. So what is wrong if you give the vaccine and the child still has maternal antibodies? What happens? I see a question on IgM. IgM antibodies for any infection show current infection. So if you get IgM antibodies, are usually the first to come up, followed by IgG, unless it's a secondary infection. If it's a secondary infection, then also IgG shoots up very fast. So somebody tell me, yes, Mohammed Nurani, you are correct. If somebody has a child, say a child who is 6 months, 10 months, still has maternal antibodies. And remember, these maternal antibodies cross the placenta. They're not being passed in breast milk. They are proteins, so they'll be destroyed in the gut. So they're being passed in placenta, and depending on the levels that were passed, they may last up to 18 months. So if you give the vaccine when the antibodies are still present, the vaccine may be neutralized because it's a live attenuated vaccine. It may be neutralized by the antibodies, and so it will not take. And we see that in up to 15% of babies at 9 months. That babies at 9 months, 15% will still have maternal antibodies in sufficient amounts to neutralize the vaccine. So it means if you're immunizing 1 million babies, and in Kenya we have about 1 million to I think 1.5 babies every month, it means out of the million, 150,000 will not, if you're assuming you give all of them the vaccine, 150,000 will not be immunized. They have received the vaccine, but they are not immunized. And that's why we are repeating at 18 months to make sure that if somebody missed or if somebody, the vaccine did not take the first time, then the second time there's a better chance, up to 95% of the vaccine working. So if the vaccine had worked the first time, then this is okay. It still doesn't harm to have a second one. But if for the many children, the 15% who may not have had a successful immunization, then the second dose gives them that chance. And you can imagine we are missing every year, say 15% of children, say 150,000 every year. In 5 years, that is 750,000, which is a very large number of people who are susceptible. And that allows for the measles vaccine to spread very easily. And that's why we say for measles vaccination, the target vaccination currently is actually set at 90%, but it should actually be higher to eradicate the vaccine, the illness. Reading now questions to the effect of, so the effect of F protein on cells is similar to the effect of H protein on RBCs? No. F protein is a fusion protein, H protein is a agglutination. The H protein in viruses is used to attach, but in the lab we use it for diagnosis because it agglutinizes RBCs. It doesn't agglutinize RBCs in the body. So F is for fusion, it helps the virus in spreading, but H is for attachment. Vaccine is will be attacked like an infection. That's a response, I agree. They want to produce antibodies to the rashes in the form of both macules and papules. There are small differences between the two, yes. You can look at the lesions we see with skin lesions from papula or pinpoint red dots, as I would describe them, all the way to pox or bullae, which could be different. So all this, and I'm sure you'll cover this in dermatology, you can have very different variations of lesions of the skin. So the vaccines given at birth still maternal antibodies. No, we don't give measles at birth. We give, most of the vaccines given at birth are, many of them are actually not live attenuated. Some may be, but most of them are not. Why do people still get the virus? And then the other thing is we give babies at 6 months because also if a baby, for example, is HIV exposed, they may not even have sufficient antibodies. So instead of waiting until 9 months, you might lose some of them, you give them earlier. In the West, measles vaccination is done after 12 months because for them, measles is less common. For us, we don't want to wait until 12, 15 months because we will lose some babies to measles. So we start earlier. And by starting earlier, we are doing a trade-off. You're trading off that those who are not immune will be immune earlier, but those who have antibodies will probably not be protected until you give them the second dose. So it's a trade-off. That's why some countries give it earlier than others, depending on the severity, on the prevalence of